Good afternoon. Thank you very much for the opportunity to present here. My name is Chris Wetzel. This is about single cells and how to isolate them. So if you're looking for single cells, we have more than 20 years experience in laser microdissection. Now that's a way how to isolate cells in tissue. If you're looking with a laser, if you're looking at liquids, that's a bit more of a challenge. So with a laser, that will be a little bit like Star Wars underwater. So there must be different techniques on how to do that. There's is also about cell sorting. No, obviously, it's not only about pairing, but it's that how to find cells and put them into the same category. So the first step is how you identify the cells. There are manual ways how to do that. The next step is aspire the cell. And there we use a capillary, so that's the replacement of the laser underwater. And the next step, obviously, is to deposit the cells and in separate wells so that it can be well identified later on. So everything is done under microscopic visual control. So it's capillary-based single-cell sorting, and we call it the MMI cell selector. The several steps are that first you have some suspension on the slide. The next step is then you have a capillary that aspires the various particles, cells. So usually in a suspension you have two sorts of cells. One sort is just freely moving, and the other sort is somehow attached to a bottom or to other things. And there is a way, the capillary, there's safety adjustment for the glass capillaries, adjustable spacing. There's also an adjustable pump volume. Typically, the volume range is in the nanoliter range. And there is a way on how to attack also those that resist the approach by scratching it, turning around the capillary and scratching it off the bottom. Then the next step is you take the cells from the donor slide, and they are then placed on a different slide, an acceptor slide. That can be many things, usually consisting of several wells, clearly separated, defined, which particles go where and how many into which well. And this is the MMI Selector Plus, so cell sorting, and it's one cell, 1 microliter, and one solution provided by MMI. And if you're interested in details, I'll be happy to show you the system at the booth number E2. Thank you very much.
Thanks, Chris. I think you get the prize for the best graphics this morning.